The Physiology of Obesity from Mechanisms to Medicine series started a few years ago when myself and Dr. Peter Aldiss at the University of Copenhagen felt that there was really scope to develop a symposium for early career researchers, which would give them opportunity to disseminate their research and bring together the next generation of leaders in obesity and metabolic disease. Now, after 3 successful symposia, first at the European Congress of Obesity, then at Experimental Biology, and a satellite meeting at Physiology 2019, we're delighted to have this webinar series to give early career researchers the opportunity to present their work, network, when at a time when the opportunity to do so is limited by the pandemic. With over 2,000 registrations so far and across 40 countries around the world, it's great to see many of you here again today. For the first half of this series, Pete and I have altered the chairing duties, and I'd like to welcome Dr. Nicole Morrissey, a postdoc researcher at the Neurobehavioral Genetics at the Mammalian Genetic Unit at the Medical Research Council in Harwell. Nicole is interested in the neural and glial regulation of behavior, rhythms, and metabolism. And before I hand over to her to introduce our speakers today, I'll just go through a few housekeeping rules. Both of our speakers will give a 20-minute presentation, and then we'll have 15 minutes for Q&A. You can submit your questions through the Q&A button at the bottom of the screen, and we will direct the questions to the speakers at the end of both talks. But you can submit these questions at any time. They can be submitted anonymously. Alternatively, you can provide your name and institution, and we will read that out as part of the question. We'll try and get through as many as possible, but we'll also try and keep to time. You can upvote these questions if one is more of interest to you, and that will mean there's a greater chance of it being asked. Please note that today's webinar is being recorded and will be available to watch again via the Society's YouTube channel. And all that remains for me is to thank the Physiological Society, Nicole, and both of our speakers today. Nicole?
All right, thank you, Joe. So it is my pleasure to introduce Dr. Gitta-Lee Sarkar. So Gitta-Lee is now a Novo Nordisk postdoctoral research fellow at the University of Oxford, where she is studying or investigating the neuroimmunology of obesity. Gitta-Lee did her PhD, however, at ETH in Zurich, where she investigated the long-term impact of maternal obesity on the brain, behavior, and metabolism of the offspring. So I'm very excited, and I'm sure you are too, to listen to Gitta-Lee discuss her research today. Over to you.
Thank you, Nicole. And thanks for this nice introduction. Let me share my screen first. So first of all, thanks a lot to the Physiological Society and the organizers for this invitation. Today I'm going to give a talk entitled The Impact of Maternal Overnutrition on Metabolism and Behaviors Across Generations. Just a bit of background about myself. I did my bachelor in medicine and surgery back in Bangladesh in Dhaka Medical College in 2009. So after completion of my medicine studies, I wanted to pursue a research career, and therefore I moved to Germany, where I did my master's in neuroscience from University of Bonn. Then I moved to Switzerland in ETH Zurich, where I completed my PhD in neuroscience in the lab of Professor Christian Wolfram. Currently I'm working as a postdoctoral researcher as a Novo Nordisk fellow in the lab of Professor Ana Domingos in the University of Oxford. In today's presentation, I am going to show the data which I have done during my PhD work. So I will start with a short overview about the background of my project. And we are in the period of big fat crisis. We all know that obesity increases the risk of several non-communicable diseases. For instance, type 2 diabetes, cardiovascular disorders, several cancers, and mental health related disorders. And this leads to premature death, and it creates a huge economic burden to our society. Over the last 30 years, the prevalence of obesity worldwide has shot up. And if we look at global obesity prevalence, we can see that the obesity prevalence has tripled between 1975 and 2016. And an interesting observation is that the women are more affected than men. And if this trend continues, the world population, obese population will be almost 1 billion by 2030. So why is this happening? We know that obesity results from the constant imbalance between energy intake and the energy expenditure. In our modern world, easy accessibility of cheap, palatable, processed calorie-dense food has increased our daily calorie intake. On the other hand, a lack of physical exercise and sedentary lifestyles have reduced our energy expenditure. And these two factors actually create an obesogenic environment. On top of that, we have genetics, which play a key role to determine which individual will be susceptible to this obesogenic environment. However, these environmental factors and the genetic factors alone cannot explain the dramatic rise of obesity in recent decades. And therefore, the concept of early life programming has gained wide attention in obesity research. So what is the ArtLife programming? So this is the process by which environmental insult such as diet, stress, infection during the critical period of development induces adaptive changes in the growing fetus that leads to irreversible physiological changes in the form of various diseases later in life. And these changes can even be transmitted across multiple generations via non-genomic epigenetic inheritance. In fact, several human and animal studies have shown the link between escalating growth of maternal obesity or overnutrition and the development of obesity and metabolic syndrome in offspring. A handful of animal studies have also showed that some of these metabolic traits can be transmitted into the subsequent generations. However, most of these studies are confined up to the second generation. But for a true transgenerational inheritance, it is highly important to show that this altered phenotype can be transmitted to a non-exposed third generation. In addition, the underlying molecular mechanisms by which these traits are transmitted across generations are still not very clear. On top of that, so far, very little studies have been conducted to see the impact of early life insult, especially dietary insult, on offspring's brain and behavior. Therefore, in this project, we aim to explore the long-term impact of maternal overnutrition on offspring's brain behaviors and metabolism. And therefore, we conducted a series of studies. For instance, we first investigated whether maternal high-fat diet can program the hedonic behaviors and metabolic states in the immediate offspring, and what are the underlying neurobiological mechanisms. And as we observed altered hedonic and metabolic phenotypes, in the next study, we explored whether these are transmitted across multiple generations. And then we also wanted to, we investigated several epigenetic markers that can be responsible for such transgenerational transmission. In addition, we also investigated whether maternal high-fat diet exposure affects the cognitive development and learning and memory related behaviors across generations. And finally, we also identified the critical windows of development that are more vulnerable for maternal high-fat diet insult. So as you can see, all these study data have already been published, and some of these works were highlighted in journals and gained wide media coverage across the world. In the interest of time, today I'm going to show some of the data from the first 3 studies. So initially, we explored whether maternal high-fat diet exposure in mice affects the brain, behaviors, and metabolism on the immediate offspring. And for that, we exposed female mice to high-fat diet for a period of 9 weeks. That is 3 weeks pre-conception, 3 weeks during gestation, and 3 weeks during lactation. As a control group, we exposed the dams to chow-fed diet for the same duration. The resultant offsprings from both high-fat diet fed dams and chow-fed dams were on chow diet since weaning throughout their life, unless otherwise specified. And then during adulthood, we characterized their behavioral and metabolic phenotype, but we found that the offspring born to high-fat diet exposed dams, we named them as F1, that is first generation high-fat diet offspring. So these offspring showed increased preference to sucrose and high-fat diet in the fat preference and sucrose preference paradigm. They also showed enhanced sensitivity to psychostimulants, for instance, amphetamine and cocaine, as well as for alcohol, which indicates addictive-like behaviors in the high-fat diet group. In case of metabolic phenotypes, we observed mild obesogenic traits in the F1 high-fat diet when they were fed only chow diet. However, when we exposed them to a junk food challenge test where they got high-fat diet, sucrose solution, in addition to water and chow food, the animals showed prominent obesogenic traits. Next, we explored the underlying molecular mechanisms. For this, we analyzed the dopaminergic circuitry in the brain reward system. The reason of focusing on the reward circuitry is that this system regulates our motivation to palatable food intake, even in the absence of energy need. In addition, this reward circuitry is also responsible to elicit drug-related rewards. And there are ample of evidence that shows that obesity and drug addiction may share common properties and common circuit level adaptation. So what we found that the offspring born to high-fat diet exposed mothers showed lower expression of TH, which is the rate-limiting enzyme of dopamine synthesis in the ventral tegmental area. In keeping with this, they also showed lower dopamine level in the VTA and the projection sites, that is the dorsal striatum and the nucleus accumbens, which is also known as ventral striatum. So overall, this shows that maternal high-fat diet exposure induces a baseline hypodopaminergic reward circuitry that might explain the observed addictive-like behaviors and the obesogenic traits in the offspring. So after these observations, next we wanted to explore whether these altered traits can be transmitted into the subsequent generation. And for this, we followed the paternal lineage because we wanted to exclude maternal confounding factors. So to generate the second generation F2 cohort, we mated the F1 male born to high-fat diet exposed dams with naive females. Similar way, for a control group, we mated F1 male born to chow-fed dams with the naive female mice. In the same way, we also generated this third generation offspring by mating F2 male from both groups with naive female mice. It is important to remember here that F2 and F3 offspring were on chow diet since weaning until adulthood, similar to their fathers. So we then characterized their hedonic response to natural high-fat diet and sucrose. And what we found that unlike their fathers, the F2 and F3 high-fat diet offspring did not show any difference in high-fat diet and sucrose preference. But they could recapitulate the addictive-like behaviors to drugs of abuse similar to their fathers. For instance, what we observed in the second generation, the both male and female high-fat diet offspring consumed more alcohol at different concentrations in the alcohol preference test compared to the control groups. In the amphetamine sensitivity test, we found that high-fat diet and control offspring showed no difference in their basal locomotor activity or the locomotion following saline injection. However, we could see that the high-fat diet offspring, both female and male, showed enhanced locomotor activity following an amphetamine challenge compared to the control littermates. Next, we also looked at their response to cocaine in a condition-place preference test, and we can observe that both control and the high-fat diet group showed spent more time in the cocaine-paired compartment compared to the saline-paired compartment. And among these groups, we can see that the high-fat diet offspring spent more time in the cocaine-paired compartment compared to the control group. So together, in next we looked at the third generation, and here we found a sex-specific effect. So we can see here that only the female high-fat diet offspring showed significantly higher alcohol preference as well as enhanced sensitivity to amphetamine and cocaine. So together, this indicates that the maternal high-fat diet induced altered addictive-like behaviors, especially for drugs of abuse, can be transmitted up to the third generation via paternal lineage. In case of metabolic traits, we observed that the high-fat diet, second generation high-fat diet offspring can develop the obesogenic traits even they were only exposed to chow diet. And we can see that the traits are prominent since weaning until adulthood. And this weight gain phenotype was associated with increased adiposity, impaired insulin sensitivity, and altered lipid profile. On the other hand, in third generation, we observed again a sex effect where the obesogenic and metabolic syndrome-like features were only prominent in the male high-fat diet offspring compared to the other groups. And this suggests that the obesogenic traits can be persistent up to the third generation on chow-fed group. Then we also checked into the dopamine level of these animals, and we observed that the second generation offsprings showed lower TH level and lower dopamine level similar to their fathers. In the third generation, again, we observed a sex-specific effect where only the females showed hypodopaminergic state like their ancestors. So after that, we wanted to explore how these effects were transmitted across multiple generations. And for this, we did DNA methylation assay of the sperm collected from the F1 and F2 fathers. And we found several differentially methylated regions between the high-fat diet and control group in both generations. However, we only found 6 significantly differentially methylated regions between the high-fat diet and the control sperms that are conserved across 2 generations. And this actually suggests that such small number of DMR actually suggest that probably CpG methylation may not be the major carrier for the transmission of the maternal high-fat diet-induced traits. And therefore, we next explored other epigenetic markers, and we focused on sperm small non-coding RNAs, since they have been reported to transmit paternal low-protein diet or high-fat diet-induced altered metabolic traits into the subsequent generations. And for that, we collected sperm from F1 fathers that were exposed to high-fat diet or chow diet, and then we extracted the total RNA from the sperm, and we did deep sequencing analysis. But we found that both groups have higher amount of tsRNA, which is known as tRNA-derived small RNAs, in the sperm. Between the groups, we observed that the high-fat diet offspring has significantly more amount of tsRNA compared to the control. We also observed 13 significant differentially expressed tsRNA in the high-fat diet group compared to the control group. Next, we isolated this tsRNA from this total RNA by a gel run, and we introduced or microinjected this tsRNA into the fertilized oocyte to generate high-fat diet tsRNA offspring. Similar way, we also generated the control tsRNA offspring. And interestingly, we could see that the high-fat diet tsRNA offspring can recapitulate the hedonic behaviors such as higher preference to high-fat diet and sucrose, as well as enhanced sensitivity to amphetamine, as well as higher preference to alcohol, similar to their ancestors. We also checked their metabolic phenotype in a junk food challenge test, as we can see that the high-fat diet tsRNA offspring, both females and males, can show the higher body weight, which is associated with increased fat mass, impaired insulin sensitivity, and altered lipid profile. And this was associated with higher high-fat diet consumption and higher sucrose consumption in the high-fat diet tsRNA group compared to the control group. So together, this study actually shows that 9 weeks maternal high-fat diet exposure can induce altered hedonic and metabolic phenotypes, which can be transmitted by a paternal lineage until 3rd generation. Not only that, we found that sperm tsRNA could be a direct mediator for such for the transmission of such phenotypes across generation. With this, I would like to acknowledge my advisors and the collaborators and the members of my former lab who helped me and supported me in this project. And thanks all for your attention. I will now hand it over to Nicole. Thank you.
Lovely talk. Thank you very much. I'm going to do a little clap. I'm sure other people would be clapping, if we were here in person. So questions will be coming at the end. So now I'm excited to introduce Dr. Soyoung Park. Soyoung did her PhD in biochemistry and molecular biology at Indiana University School of Medicine. She then joined the lab of Dr. Sebastian Bure at the Children's Hospital in Los Angeles back in 2014 as a postdoctoral fellow. And here her work has been examining how maternal nutrition affects hypothalamic development and metabolic regulation in mice. So over to you, Soyoung.
Thank you for a nice introduction, Nicole. I'm very, thank you so much for staying online, and thank you for invitations. I'm very excited to share my work today. Give me 1 second for sharing my slide. Okay, all right, here we go. All right, so I'm a postdoctoral fellow at Children's Hospital in Developmental Neuroscience program. I'm actually a senior postdoc. The title of my talk today is Maternal Obesity Induces ER Stress Causes Metabolic Alteration in the Offspring. Sorry. Not sure why it's not moving. Okay, here you go. Sorry. Jitari did such a great job to introduce about the introduction about the obesity and type 2 diabetes. So I'm not going to go all the details about the introductions. However, I will give you a short, brief introduction about the obesity, and then I will briefly introduce my the work that I recently published in this year. And if I have a time, I can give you a couple of slides to what I'm working on. Which is on published data. So obesity actually currently affects more than 30% of the reproductive age women, and it continues to be one of the major health problems in the state. I don't think it's only the state, I think it's a whole all over the world problem. And with the, sorry, this is weird. Okay, so with the clinical and animal study shows that exposed to an obesogenic environment during pregnancy or early postnatal life predispose the baby to develop the metabolic syndromes such as obesity, type 2 diabetes, and cardiovascular disease. And these metabolic syndromes are due to defects of the key metabolic organs such as brain, in particular hypothalamus and pancreas and gut and adipose tissue. So in our labs, and I'm actually really interested in how central nervous system control the food intake and then energy expenditure. And we all know that hypothalamus has been extensively studied to working in how they contribute to food intake and energy expenditure. And I'm not going to go over the detailed overview about this hypothalamic circuitry, but hypothalamus can be divided several different compartments, such as archaeonucleus hypothalamus, which is we call the ARH, actually contains a specialized neuronal populations, which, for example, HRP and PY neurons are orexigenic neurons. They actually stimulate the appetite. And on the other end, POMC and CART neurons are acting as a major negative regulator of energy balance by reducing food intake and increasing energy expenditure. And these neurons can be actually stimulated by several hormones from periphery, such as leptin from adipocyte, insulin from pancreas, and ghrelin from the gut. And these neurons population actually sending the proper projections to the several different compartments of the hypothalamus. For example, they're sending projections to PVH, which we call the paraventricular nucleus hypothalamus. And these projections actually can go to either median eminence or brainstem to control neuroendocrine and autonomic tones. So when I actually joined the Sebastian's lab, we are actually really interested in how obesity and diabetes can cause a cellular stress response. And one of the most expensive studies was endoplasmic reticular stress. And it has been studied at least more than 20 years. And then this is one of the pioneer work from the Oskan's group showing that the end of ER stress can be linked to obesity and insulin actions type 2 diabetes. And just remind you that ER is the site of the protein folding, and ER stress can be caused by building up all the misfolding or unfolded protein. And this is a signal cascade showing mediated through these three parallel pathways. I'm not going to go over this pathway. However, in our work later, I'm going to show you that using the certain individual markers such as ATF6, XBP1, ATF4, these markers to look at the overall ER stress in my study. And so after that, following the paper, the Oskan same group, they actually published this very well studied cell metabolism paper in 2009, showing that hypothalamic ER stress can be one of the possible mechanisms to induce a leptin resistance. So they're actually using the OBOB, which is a leptin deficiency mouse model, to look at how hypothalamic ER stress can be occurs. And then after they treating with the ER stress inhibitor to look at, which is they using the TUDCA to show that they actually decrease the body weight. So, so they actually conclude that hypothalamic ER stress is one of the possible critical mechanisms to induce leptin resistance. However, the importance of the hypothalamic ER stress during development and then its long-term metabolic regulation is not really still known. So in our lab, and I actually start to working on this project, then how hypothalamic ER stress can be developed during development month. So we're using the same OB mice model, which is leptin deficient mouse model. As all you know, the leptin deficient OB mice model become hypophagic, diabetic, and they have like higher body weight, adiposity. They're also glucose intolerance. However, when they're actually doing the pre-weaning stage, they actually not difference their body weight or adiposity compared to the wild type litter mate. They actually develop this metabolic phenotype after 4 to 5 weeks old. So we want to look at is there any like how their hypothalamic ER stress can be developed in this mouse model. So we actually using the P12, which is the time point that their actually body weight is not changed compared to wild type litter mate, and their adiposity is similar. However, when we look at the ATF4, which is one of the ER stress markers to look at the archaeonucleus hypothalamus, we do see that significantly ATF4 is increased in the OB neonate, which we conclude that this hypothalamic ER stress can be developed as early as very early during development, and is also independent of adiposity and then independence of the body weight. So we conclude that actually leptin modulate hypothalamic ER stress during development and actually has a long-term metabolic effects. However, I'm not going to go to all the detail about this paper. This paper was published this year in Nature Communication. Because like in human, leptin deficiency is such a rare, it's very rare in human, and most of obese individual and then like diet-induced obese mouse models are hyperleptinemic or like leptin resistance. So we want to move on to one of the more like clinically relevant mouse model, which is a maternal obesity. So I think that Jitari did such a great job to introduce maternal obesity, but maternal obesity is already known that associated with the increased risk of the obesity, and then induces like metabolic dysregulation in offspring, and also it persists into the adulthood, which means obesity can be developmentally programmed. So we want to test. Based on our previous work, we want to test whether actually very straightforward questions, whether maternal obesity causes ER stress during development. And second was what's the functional importance of the early ER stress in metabolic regulation and hypothalamic development. So we actually come up with our own mouse maternal obesity model. It actually consult with Dr. Sue Ozensker from Cambridge. They're actually using the condensed milk, but we actually using the high fat, high sugar diet, which is like usually Western diets, not only the high fat diet, they actually comes with a like sugar or sucrose. So we actually using the 58 kilocalorie per percent of fat with a 20% sucrose. And then I fed it, the wild type female mice were fed either chow diet or high fat, high sugar diet for 6 weeks prior to mating. And after mating until the offspring's weaning day, they're on the high fat diet or chow diet. So after offsprings, after weaning, they're on the regular chow diet. So we want to test whether this is actually working. So we find that when you look at the body weight chart, the mothers become nearly increase the body weights compared to chow diet during gestational and after mating. And also we do find that they're actually fatter, they're higher adiposity, and also there's glucose intolerance. So we know that this is actually working. So we actually want to follow up the offspring, which is most like what I actually really want to like examine. And then we do find that these offsprings are heavier compared to chow diet, and they are also glucose and insulin tolerance, and also they're fatter, and then they're using they eat more, they use less energy. So we find that maternal obesity actually induces metabolic microgram in the offspring. So I mentioned earlier about the hypothalamic ER stress, and that could be one of the possible mechanisms to read the leptin resistance. So how actually does maternal obesity induces ER stress during development. So we actually dissect the hypothalamus, and we actually confirm that hypothalamic ER stress can be occurred as early as E16.5. This is a whole hypothalamus, by the way. And then we look at the P10 to look at the arcuate and PBH area to see that most of ER stress markers are mainly expressed in the arcuate. And then we actually collect the other metabolic organs such as pancreas, liver fat. Excuse me, we don't see that any change in liver and sub-Q fat, and however, we do see some of the changes in pancreas. However, if you are a pancreas person, you probably want to know that which one is this like exocrine cells or endocrine cells. By the time when I actually do that, we just took out the whole pancreas, so it's not really hard to say that this is only from the endocrine and exocrine. This is a whole pancreas, by the way. So we confirmed that maternal obesity actually induces ER stress during development, as well as not only hypothalamus, but as well as other metabolic tissues. So to test whether the importance of the early ER stress, we actually using the TUDCA, which is ER stress inhibitor. So it's already known that this is TUDCA is the chemical chaperones, and is FDA approved chemical. It's known that TUDCA is actually cross the blood-brain barrier. So sorry about that. So TUDCA, we're using this TUDCA to inject this during the preclinical time period from the P4 and P16, and to look at what's the long-term consequences of the effects on physiological and neuroanatomical outcomes. So when I actually working on this study, and one of my friends sent me this, and they actually in Columbia University, they actually started a clinical trial with the TUDCA with a new onset of type 1 diabetes patient. I think they actually recently finished their clinical trial. Hopefully they have really good outcome. I haven't followed up their study, but during using the neonatal TUDCA actually treatment to see that I have some of the reverse expression of the most of elevated expression of the ER stress markers in the offspring, in the hypothalamus, as well as other in the pancreas, but not other markers. Again, I'm not going to look at the specific ER stress pathway. This is global overall ER stress expressions, but this was like really exciting to see that this is actually working. Our TUDCA actually reduces like not only pancreas, but also brain ER stress expressions. To test whether these neonatal TUDCA treatments has a long-term beneficial effects on body weight, we do see that these are injected neonatal TUDCA from the high-fat offspring from the high-fat, high-sugar diet mothers. They do actually have a less weight compared to the vehicle-injected offspring from the high-fat, high-sugar mother, and they also improve the glucose and insulin tolerance. So we actually put them in the metabolic chambers to look at what's the metabolic outcomes. And we do find that like neonatal TUDCA treatments actually improve the metabolic programming, such as they actually less fatter, they reduce the food intake. Also, not only that fat mass, but when we look at the adipocyte size, this is a high-fat, high-sugar diet offspring, and this is a neonatal TUDCA treatments. And when you look at the adipocytes, they actually decrease the adipocyte size, which is significant. So we do see that neonatal TUDCA treatments not only improve the metabolic program, but also they reduces the fat size. So we want to test whether actually maternal obesity induces hyperleptinemia. So I collected serums from the mother and an embryo from E16.5, and I do see that the hyperleptinemia, maternal obesity induces hyperleptinemia as early as E16.5, and also we do see that neonatal TUDCA reduces leptin level. So we want to test whether this is like what's the signaling pathway in the leptin. So we want to look at the P-STAT pathway in the hypothalamus. So this is a very exciting data that we find that maternal obesity actually reduces P-STAT leptin resistance. So leptin sensitivity, such as when you look at the P-STAT3 positive cells in arcuate nucleus hypothalamus, they actually reduces P-STAT3 positive neurons in the offspring of the high-fat, high-sugar diet. in the ARCHID specific, but not the DMH. But when we look at the neonatal TUDCA treatments, they actually increase the P-step positive cells. So we find that maternal obesity causes central leptin resistance, and then that is reversible with the neonatal TUDCA treatment. I'm sorry about my dog, is kind of like disturbing me. So anyway, I mentioned earlier that the proper projection from ARCHID to PBH is really important to control the neuroendocrine and the autonomic tones. So we want to test whether maternal obesity actually alter or alter the certain hypothalamic projections. So we can find that maternal obesity actually reduces POMC axonal projections, specifically not the HRP. But also when we look at the neonatal TUDCA, actually ameliorate this disruptive POMC axonal projection in the offspring of the obese then. So we want to test whether what actually caused this, what's the components of the inducing all the ER stress and then hyperleptinemia. So we want to go back to the diet, and we find that there's such a lot of saturated free fatty acid. So we are using the in vitro cell experiments to which free fatty acid actually directly induces hypothalamic ER stress. This is an in vitro study. So we find that this palmitic lauric mastoic acid, which is a saturated free fatty acid, actually directly induces hypothalamic ER stress, but not the unsaturated free fatty acid, which oleic. And let me skip this. So we do find that in our in vivo mouse model, we actually have a higher circulating free fatty acid model. So so we want to test whether actually what's the effects of free fatty acid on the hypothalamic axon growth. We're using the X-plan to see that how this free fatty acid impact the axonal outgrowth in using the X-plan. And we find that it's very interesting. This is a vehicle, it's control treatments. And when we look at the combination of the free fatty acid, we do see that it's impeded axonal growth. However, it is improved with the TUDCA co-treatments. So we find that saturated free fatty acid causes ER stress induces disruption of the axonal growth. So to summarize, maternal obesity causes metabolic alteration and induces ER stress in the hypothalamus and as well as in the pancreas. And maternal obesity actually impair POMC axonal growth and attenuate leptin sensitivity and relieving ER stress through the neonatal TUDCA, which is a critical time window of the hypothalamic developments, has a long-term beneficial effects on body weight, glucose homeostasis, leptin sensitivity. And free fatty acid directly blunt axonal outgrowth and from the archionucleus hypothalamus, and then TUDCA co-treatments improve the axonal outgrowth. So I'm gonna give you a very short overview about what I'm actually working on, and I'm really excited about our new data, but I'm really interested about the sugar and artificial sweeteners. So we are looking at the effects of the low calorie sweeteners on the metabolic effects and then hypothalamic developments. So I'm not going to go everything, but we're using the aspartame and ribonucleoside, which is a stevia, to look at the metabolic phenotype and as well as hypothalamic developments. And just this is, I'm going to skip, but there's no change body weight, but we do see that aspartame offspring from the aspartame fat, they actually have a higher body weight, have a higher fat mass and glucose intolerance. So acknowledgements, Sebastian's actually recently moved back to Lille. So I think he's looking for a postdoc. He's a great mentor and scientist if you're looking for the postdoc mentor. And then 2, 3 actually very talented technician I work with it, and I moved to Pet Level Lab. So, and also thank you for the funding, and then thank you for staying. Thank you for listening, and I'm happy to answer your questions. Sorry, I didn't really, rush it.
Thank you. That was 2 wonderful talks there. Just before we move to the Q&A tab, I'll kick us off while people are submitting their questions. Gitali, you were talking about the differences between the F2 and the F3 generation. In the F3 generation, if I read your slides correctly, the effects were in females only. Could you speculate as to why that was?
Well, I mean, this sex-specific effects, it's really a big thing right now, and people are really exploring what could be the reason. It's not only us, there are several other groups looking at different other stress effects, like paternal or maternal. They also saw the sex-specific differences. So we did not really explore in details in that direction, but we hypothesize that there may be several factors that can lead to these differences. First of all, the gonadal hormones can be one of the factors. Second is the gamete, male and female gamete. They have actually different time point of growth, and that time difference can affect can be affected by the time of exposure of the insult. So this could be another thing. And the other one is like placental gene expression. So that has been shown that the maternal or maternal heifer diet insult can affect this placental specific gene expression, and that can lead to subsequently affect the expression of different genes in male and female offspring. So this could be another option. So that would be very interesting to look into why these animals actually shows differences based on sex. And yeah.
And just before we move on, following on from there, did you consider an intervention arm such as caloric restriction or exercise in your studies to see if you could negate the effects in subsequent generations?
Yeah, that was something in plan, but yeah, then I finished my PhD and actually it was handed over to someone else. But yeah, that was in plan to look at, whether combination of exercise or intervention can, reverse the effects what we observed in the offspring.
Okay, so if we move to the Q&A tab, we've covered that first question there. Soyoung, if we transfer to you and your TUDCA treatment, how do you think it regulates leptin levels during the developmental window? And is it through reduction of ER stress in peripheral tissue or through a reduction in ER stress centrally?
It probably both. My injection is IP injection. It's not like central injection to the specific brain region. So I can exclude that it might be same as like periphery plus, and it also cross the BBB area. So it might have a both impact. However, I think that Oskan's group actually in that 2009 paper, they do inject it centrally in the hypothalamus. I think they do actually inject it the third ventricle. So it's not like specific area of the hypothalamus, but the third ventricle to see that reduces hypothalamic ER stress. So, however, I think in our study, I can say it's only central or peripheral. It's my work was like more like IP injections. It's a global impact. So yeah, but that's actually great patients. Yeah.
Young, I've got a question. So you were looking at markers of endoplasmic reticulum stress in the hypothalamic regions, and you saw an increase in these markers in the arcuate hypothalamus compared to other hypothalamic regions. I was just wondering if you looked at other non-hypothalamic regions that are involved in appetite control, such as brainstem or maybe more forebrain regions.
Yes, I actually did. That's such a great question. We actually thought about this like more like hypothalamic specific ER stress like response. So we use cortex as a control, which is non-really metabolically related the brain area, I guess, and then it didn't really change anything. So we do see, like we only have a cortex. I think that I haven't looked at the brainstem, like DMH area, DMB area, but in Edinburgh, when we compare the cortex and hypothalamus. It was like more hypothalamic specific expressions, but I haven't really looked at the hippocampus or any other brain region, but as my control was more specific in the hypothalamus. That was actually a really good question. Yeah. We actually did some fact sorting using POMC and MPY-GFP mouse model to look at whether this is another thing, that whether this is like really specific into the specific neuronal cell types, because our study was only changed in the POMC axonal projections, but not the HRP. But we do see that similar changes in only specific in POMC HRP mice model. Yeah.
Thank you. So I'm going to move on to some of the questions in the Q&A section. So this question is from Sam Oakley, and it is a question for Dr. Sarkar. So, it's linking both the talks. As Dr. Park found that it was primarily the saturated free fatty acids that cause the ER stress, do you think that you would have found the same result in your studies if the high fat diet was low in saturated fats and high in unsaturated / polyunsaturated fats?
Yes, I do. Yes, I do. When we actually do the in vitro study, so I actually using what's the sugar effect. So we, in our diet, we have 20% sucrose and with a lot of like fat with a saturated fatty acid. It's not, I didn't show, but I actually test individual free fatty acid and then with a combination and with sucrose or without sucrose. I don't see that any sucrose effect to induces directly hypothalamic ER stress markers, but only in the fat, like free fatty acid. Yeah.
Gitali, do you have any anything to add to that question?
If you I think it depends, right? So the dietary insults that we are using in the diet, different diet models, it actually varies with the duration of diet and then the ingredients that are using. For instance, in my case, it's just the high fat diet, and we really didn't go in details of looking at which specific diet has, like dietary ingredients has this effect. So obviously it varies with different dietary components, and there are studies where only the sucrose, high sucrose diet could show that they have the metabolic syndrome-like features, right? So yeah, it's like a huge area, and different dietary models on different time points of exposure could create different levels of phenotypes in the models.
I also have another question for you, Gitali, if that's okay. So when you were looking at the VTA, the ventral tegmental area, you were looking at tyrosine hydroxylase expression and also dopamine expression. Now I'm not sure what other enzymes are expressed in the VTAs. It's not an area I've really studied myself, but I was wondering, so downstream in that synthetic pathway, you've got, what is it, dopamine beta hydroxylase and other enzymes for adrenaline and noradrenaline. I was wondering if when they are expressed in the VTA, and if so, do you look at those levels as well, or maybe inputs from other brain regions?
So for this study, actually, I didn't really look into the downstream synthetic or the degrading enzymes, but what we looked of the downstream substances in dopamine signaling is the dopamine transporter and the receptors, and we could see that there is a low expression of dopamine transporter in VTA, and at the receptor level we saw the lower expression of both dopamine 1 and dopamine 2. So the dopamine 1 was lower, but dopamine 2 was highly expressed in the projection sites, that's in the ventral and dorsal striatum. But obviously the other markers like transmitters, for instance, norepinephrine, epinephrine, and those markers could also have interaction with the dopaminergic signaling. But yeah, obviously we did not really go into looking for those interactions in those regions. But that's a good question.
And Soyoung, just before we finish, I was interested, as was Sam Oakley in the question and answer tab, in that preliminary data that you showed at the end with aspartame. What do you think the mechanism for that is that's driving the glucose intolerance?
That's I'm actually trying to find out. So we do have a in vitro study. It was published last year in Frontier Endocrinology, that I tested a bunch of the sweeteners, like whatever is out there in commercially available. I used to see that whether these sweeteners directly impact the axonal growth. So we do find that one of the sweetener was really impacted, but not the aspartame. So it was kind of like, I don't know. And when we actually looking at the aspartame consumptions, and then we follow the offspring, these offsprings are actually really become like metabolic dysregulations, and they do actually have a hyperleptinemia, and also as well as they actually disrupt the hypothalamic feeding circuitry, and also they are they actually change the beta cell mass, which I didn't show you. So I was looking at the pancreas at the same time. So I wasn't really 100% sure right now, but I think they're because of their higher fat mass compared to their other offspring. I think this is like because like, and also there are like bigger fat cells in which I didn't show you, but I guess that there's like that's why, because they're higher fat mass, they actually maybe drive to hyperleptinemia maybe, but also they're also glucose intolerance, such as they also have a lower beta cell mass in the pancreas. So then maybe they kind of like try to make a link, but also I'm really interested about the hypothalamic inflammation. They are actually showing it shows that they actually have a higher inflammation in the hypothalamus, specifically in this offspring from the aspartame fat dams. So it's very exciting. I'm not really, I shouldn't, it's still ongoing, but stay tuned. I think we're gonna submit papers soon this year. So hopefully you guys can read it, yeah. But this is really exciting. I think that sweetener is a really exciting area.
Yeah, I agree. We'll look forward to reading those papers. Well, all that remains is to thank both of our speakers, my fellow chair, Nicole, and the Physiological Society. Thank you, everyone, and we'll see you again soon.
Bye-bye.
Thank you. Bye.